Hello and welcome everyone in our very special episode of Researcher Celebrities. Today we are fortunate enough to have Professor Suresh Subramani, who is what we can say is the torchbearer in his field of research. And I would like to welcome Sir for giving us time, which we can see that because of the responsibilities, it is a lot of time crunch you are always in. So thank you very much, sir. And I would like to welcome you.
Thank you.
So first thing what we will do today is we will ask Professor Subramani that how he started as a student, and then with the experience of over a decade or 5 decades, I will say, where starting from his bachelor's to being the vice chancellor and distinguished professor at University of California, how he have traveled this. So, sir, brief about.
Well, first of all, thank you, Raja. Good morning to everyone. And thank you so much for the opportunity to talk in particular to young scientists who are shaping their careers at the early stage. And I hope what I will say will be of some use to them. So since you asked me to summarize close to 50 years of my past history, let me just begin with when I got interested in biology or biochemistry. Specifically, I got very interested in biochemistry at a very early age. In high school at that time, I was influenced by two things. One was that I had an uncle in the family who was in the hormone research lab as a faculty member at UC San Francisco. And I was very impressed by his description of what science was about. And then I also had some fantastic chemistry and biology teachers in school who inspired me to go into science rather than into other disciplines. So right after high school, another very significant event happened, and that is that I applied for something called the National Science Talent Scholarships. I had no clue, in fact, that I would be even eligible for this thing, but I got this thing. And so, as you may know, they pay for your education all the way from your bachelor's degree till the completion of a PhD, assuming it was all done in India. So because my family was a middle-income family, we were 5 children, the fact that one person's education was getting taken care of meant a lot. And it also meant for me, that gave me the freedom to pursue whatever it is that I wanted without feeling that I was being a burden on my parents or my family. And so it opened my eyes to the scientific excitement and to scientific heroes, because my cohort of students consisted of other scholars for mathematics, physics, chemistry, and biology. So we were all together in one cohort. It was also the excitement of doing research and pulling all-nighters for experiments, and we would talk excitedly about our results, and we would be watching over instruments as the data came out from scintillation counters or other machines. And mostly the thing that excited me was the fact that it gave me intellectual freedom to dream about and play with scientific puzzles that really fed my curiosity. So that was, this freedom was the biggest aspect of this excitement. So because my path was decided mentally till my PhD, I joined Ferguson College in Pune for my bachelor's in chemistry. Had some great mentors there, and then transitioned from there to go to IIT Kanpur for my MSc in chemistry. And at that time, it was one of the best chemistry departments in the country, with some fantastic faculty, and only 15 of us in that cohort. And they paid so much attention to us that if we didn't show up for class one day, the professor would be in the dorm saying, "What happened to you?" And there was even a time when everyone in the class failed in some quantum chemistry, and we complained that the head of the department came over and asked what was the problem. And we said, well, we don't really understand the teacher. So they started afresh, with a best teacher for that. and we restarted the course. And so that was the kind of attention we got, very personal and attention from great scholars. So after I finished my master's, then it was an opportunity to do my PhD, and I had the opportunity to go to the Weizmann Institute in Israel or to UC Berkeley. And initially, because of proximity and the fact that my uncle had actually studied a little bit in Israel and spoke very highly of it, I thought I would go to Israel. But in those days, you needed a separate passport for Israel or South Africa. And that process was delayed by quite some time. And so in the end, because I could not get a passport to go to Israel, I ended up coming to Berkeley, and I don't regret it one bit because it was a fantastic time. And because I came into a biochemistry department, having done chemistry and zero biochemistry at that point, I felt that the easiest transition for me would be into structural biochemistry. So I worked in the laboratory of a renowned scientist called Howard Schachman, who was a physical biochemist, and studying protein-protein interactions in bacterial enzymes and looking at allostery and so on. And then after I finished in 4 years, I decided, of course, that I would look for a postdoctoral stint. And I thought that I was interested in neurobiology. And in fact, when one of the people from Caltech came to give a seminar, I asked him if I could do a postdoc, and he said, sure, you can come on over. And then I still remember this conversation I had with my postdoc, about my PhD mentor, Dr. Schachman, where he said, you should be thinking about a postdoc. And I said, yes, I've already started, and I think I'm going to become a neurobiologist. He was very wise. He said, look, there are two ways in which you could decide upon your future career. And I said, what are those? And he said, well, one, you do as you just did, pick the best person in a particular field and go work for them. Or he said, don't worry about the field, just pick a fantastic person. I said, like who? So he said, like Paul Berg at Stanford. And I said, why would he take me? Because, he's studying viruses and tissue culture cells and so on. He said, let me talk to Paul. And so on Monday, that was on a Friday, on Monday, I got an interview to go talk to Paul Berg. And I was very uncertain that he would take me. So I kept asking these questions like, if I were to come to your lab, could I do this? Or if I were to come to your lab, would this happen? And after a little while, he got a bit irritated. He kept saying, you keep saying, if I do this and if I do that, I thought you were coming to the lab. And I said, look, I said that because I was just not sure that you would take me. And he said, no, it's all decided. You're coming here. So that's how I ended up in Paul Berg's lab. And that was between 1979 and 1982. And those in the audience who followed history will know that this was very exciting times during my stint in Paul Berg's lab, because this was when the transformation procedures for getting DNA into mammalian cells was being developed. All the selectable markers were being developed, and I was involved in developing one of these selectable markers to get DNA into mammalian cells. Introns and splicing and promoters and enhancers, these were all being discovered at that time. And I couldn't have been in a better place at a better time because in 1981, during my stay there, Paul Berg got the Nobel Prize for his work broadly in nucleic acid chemistry. So when I started applying in the fall of 1981 for jobs, it was a fairly easy thing. At least I got to the stage of an interview at several places that I applied to. And I had multiple offers, but my wife and I decided that many of the places I had offers at were very, very cold. And so we decided to come to UC San Diego in 1982. So it's now 40 years since that point, and I've had a fantastic career and opportunity. So I want to just highlight for the young people, how truly blessed I have been in terms of having an unusual and unanticipated opportunities thrown at me all along. So basically, as long in looking back, I felt like these opportunities come in everyone's lives, and you just have to be aware of it and open to the possibility and grab it when it comes. And so let me just give you some examples of that. So initially, when I made the switch from chemistry to biochemistry, I thought during my undergraduate days I'd end up doing something in chemistry, right? So I switched to biochemistry, and then in Paul Berg's lab, I switched to molecular biology. And eventually, for the last 30 years, I've been a cell biologist. So what this has taught me is to avoid labels, so you're a biologist or a scientist, and focus more on acquiring whatever expertise is needed to solve a problem. And this is what I've communicated to the 100 or more people that I've trained in my laboratory over the period of time. These include undergraduates, PhD students, and postdoctoral fellows. Right. The second thing is that my entry, particularly into cell biology, was completely serendipitous, meaning that we happened to be working with, we cloned with Don Helinski and some students here, the firefly luciferase gene, which has now turned out to be a really widely used reporter in all of biology. And by chance, because I happened to be next to a lab doing cell biology, we asked where this protein was and found it was in peroxisomes. And so when I applied for my first NIH grant, I got a terrible score. They basically said, this guy is not a card-carrying cell biologist. He knows nothing about cell biology. And in fact, they said that he doesn't even know that yeast have no peroxisomes. They were drastically wrong. And we've made a living out of that for the last 35 years. But the good news is that from a completely unexpected turn of events and a chance observation in my lab, 35 years later, I just got renewed for my grant that I've had for 35 years on continuing my work on targeting of proteins to peroxisome. So this is just an unexpected event, right? Then around 1987, just 5 years after I started my career here, I started having these dual opportunities for my career. That spanned academia on the one hand, where I was, and industry on the other. So it started with my being a consultant and on a scientific advisory board for some local companies, as well as a company in India called Astra at that time in Bangalore. And so Astra in Bangalore asked me to come and visit, and so every year I would go there and be on the scientific advisory board. And then this provided me with the opportunity from 1996 to 2001 to actually help a US company here called Affimax, based in the Bay Area, get their work done in India, in Bangalore, through Astra and through a company that's now called Syngene, which is a part of the Biocon group of companies. And so from that time in 1996, I was 50% in academia, 50% in industry. And this was unusual because, most universities would have said, sorry, if you want to be in industry, just go. In fact, we were ready to leave, and I had told the department I would resign. And then it turned out that they were open to the idea of a 50-50 appointment because there was no conflict of interest. And so I worked with Affimax for 5 years. And during that time, they got acquired by GlaxoWellcome, which then merged with GlaxoSmithKline. So Affimax became a part of GlaxoSmithKline. And then in 2001, they were not sure which way they wanted to go, what they wanted to do with the group in India. We had about 30 people working in India at Syngene on their projects. So I backed off of that and decided at that time that since it was fun doing basic science and science management, that I would turn my efforts internally to helping pay back to the university, which had really helped my career along the way. And really, I was exceptionally happy and in deep gratitude over the fact that they'd allowed me to have this dual career. So that became a pattern then. From 2001 to 2016, I was involved in a variety of academic positions, starting from chairman of the department to associate dean to dean, and then associate vice chancellor, and then the executive vice chancellor. And in all of this, my only condition was that I'm happy to help the university with only one condition, that if you ask me to stop my research work in the lab, then I'm not going to take the job. So again, the university was very flexible in the fact that I had at times 2 or 3 jobs, but always the anchor was my lab that kept me going. And then I just put in the extra time that was needed to help run the university. So particularly during my stint as the executive vice chancellor, for those of you who might not know, this was basically running the whole university on the academic front. I was in charge of all of the undergraduate and graduate programs on the campus, as well as the hiring and promotions of 2,000 faculty. And I indirectly oversaw an annual research budget of $750 million. So it was a fantastic experience because I jumped in at a time when the University of California had the most severe budget cuts. And this gave me an opportunity to help put the ship back together again with a lot of help from a lot of people. And I was really happy to see that in this time, our rankings have gone up. Our Division of Biological Sciences is ranked among the top few in the world at this point in time. So, but I just want to digress a bit because this is how life has its twists and turns. When I applied for a PhD, I actually applied to UC San Diego, and I was rejected at UC San Diego. And fortunately, I went to UC Berkeley. And then, some 30, 40 years later, I became the chief academic officer of the university that had rejected me for their PhD program. So I'd love to tell the story, particularly to the incoming PhD students every year. And they always enjoyed it. So, so finally, just to end this thing, in 2016, I stepped down from being the executive vice chancellor because there was a phenomenal opportunity that fell into my lap for doing something for my country of origin, India, of course, and doing something significant to help science and technology there. And this opportunity came about, as you know, because of a very visionary philanthropic gift from the Tata Trust in India, which then required the creation of 2 institutes called the Tata Institutes for Genetics and Society, one in UC San Diego here, and the other one that I created in Bangalore from scratch on the campus of the NCBS and INSTEM in Bangalore. And so I stepped down from being the EVC to becoming the global director of this entity called TIGS. And it was created as a not-for-profit research and development organization focused on research and training in the use of advanced genetic technologies to tackle healthcare security and food security, particularly in the Indian context. And today, TIGS India has about 70 people that I recruited. And so that's where we are now. So it's a long-winded answer to your question, but that's how I got into science. And if I think back at how I got here, I get goosebumps because none of it was planned or anticipated, and it just was taking advantage of opportunities that came along the way and helped by lots of people that I can't possibly thank at this point in time. But I just want to end with saying that it's been a special privilege and a pleasure to not only train over 100 people, but learn from them and interact with them and watch their careers grow. Because there's an Indian saying that, a student has not completed his training until he or she can become a guru. So in other words, you start off as a teacher, and when you come to the point where you learn from your student, that's when you completed your job. And I feel proud that many of these students are doing exceptionally well around the world. So anyway, so that's my answer to your first question.
Thank you. Thank you very much, sir, for sharing this valuable thoughts and experience which you walked us all through. And here, I just want to take the liberty to add one thing that Professor Subramani just narrated the entire story. And we were hearing that he was fortunate enough, he was like shifting the fields and all that. But let me tell you, there is no shortcut for hard work. Okay, he was doing his part of hard work, and he is humble enough to say that it just happened in two places, but it is just his hard work, which he has put when he was a student. That is the live example of that, is like why he got the funding for doing all his education. And when he, as a student, he did this. As a mentor, he did what I think this will be another episode, that what his students are doing, what his mentees are doing as a mentor, what he has done. So I would like to mention, as you said, about a student is not completed till he has teach and learn from the students, his students. There is a very famous saying of Albert Einstein, who said, information is knowledge. The only source of knowledge is experience, and you need experience to gain wisdom. So what Professor Subramani has shown us is like all of these makes a good complete circle, and you have to do that. There is no shortcut. And I will say this 1,000 times to all of our researchers: be humble, do your hard work, be humble in there. So now when we talk, whatever we have heard with Professor Subramani, that over the 4 decades of the experience of academics, management, administration, industry, whatnot, I cannot think of anything which people can count as a researcher in any of these fields. But you have enjoyed your research to that power that whenever there was possibilities of doing anything more, but you anchored everything to the research. How different was doing research in different fields?
Yeah, so firstly, my inspiration for research actually came, as I said earlier, from having great teachers and the scholarship that it really was provided by the government, the National Science Talent Scholarship, which basically encouraged me to, said that someone was willing to invest in me when I was 18 or 19 years old, to say, you know, we are happy to pay for all your expenses till your PhD. This is just unbelievable, you know, even when I think about it. And for the government to say something like this was just fantastic, right? So that basically meant that as long as you do put in the hard work, as you said, you know, the government is going to help you get a PhD. And that's what this was. So it also gave me the opportunity to meet fellow students from all different fields, from chemistry, biology, physics, and mathematics. And every summer we were sent to a national lab, initially of the choice by the government, but once you became a master's student, I could pick any lab in India to go to and spend the summer there. So this exposed me to the excitement of research in a variety of different labs, and also gave me this freedom to operate. As I said, I could pick a lab, I could work on any problem and learn. And you know, as you know, most scientists don't have fixed hours. You can spend all night with your experiments or go in whenever you want. So this was just tremendous, that being your own boss really meant a great deal to me. So, but importantly, I learned that the fun of doing science was not just about me. It was about the teammates, the interactions, the opportunity to learn. And I cannot overemphasize the fact that it is a lifelong opportunity to learn. You know, so I learned biochemical pathways when I was doing my PhD at UC Berkeley. I'm relearning all of those metabolic pathways now because my research is driving me into that, right? So you're constantly learning new things. And if you follow the problem and go to whatever field it takes you to in terms of the tools necessary to solve the problem, that means then that I've learned along the way bioinformatics, I've learned genomics, I've learned so many fields along the way, right? So this means it's a lifelong learning journey. And also, I've learned that it is important to walk along untrodden paths. So you can follow someone else's thing and follow the hot fields, but I've always felt like we should branch off into things that are completely unknown to the world and unknown also to me. And by paying attention to these unexpected side adventures, that has kept us going for the last 35 years, as I said, particularly now work on cell biology. I never trained as a cell biologist. In fact, I have a document from the National Institute of Health saying I'm not a card-carrying cell biologist, but yet here I am. And finally, I just want to say that I've learned to always follow the question and the problem and be less enamored with the tools. So some people might be, let's say, an X-ray crystallographer or NMR specialist and so on. So then they use the tool to say, what new problem can I solve with this tool? That is one approach to science, and there are people who become successful with it. But I find that I'm more interested in the problem and asking the right question, and then going and finding the tools wherever they exist. And of course, you cannot have too much of an ego in this, because you have, while you're being a mentor to a student, you have to be able to tell your student, look, I don't know anything about biophysics. Your problem needs biophysics. I can put you in touch with a friend who's a biophysicist, and together we will learn, right? So this is the opportunity to learn from your students as you go along, because they're the ones who are breaking new ground and bringing their resources and talent into the equation. So it's a constant exchange of ideas and experiences and information. And this is what has created the joy of teaching and learning all through your life.
So how what you mentioned essentially is the evolution of a researcher from student to a mentor. And you rightly said that there is, like, you were always ready to take chances. If I would have to ask you that taking chances, because you took chances as a student, as a PhD student, then as a faculty and everywhere, but giving the present scenario, because you know research way more than most of us, our audience knows, is there any good checkpoint or time point when they should be willing to take more chances, like when they're a PhD student or looking for postdocs in their lab, choosing for faculty? And because nowadays, joining academia or industry, it is like so many people get diverted and they make their minds, and some of them still keep research as the primary thing. So how do you see that when is a good time to take chances?
Okay, so I think you have to learn early to take risk, and you must be willing to take risk all through your life. But having said that, what you learn is how to mitigate risk and how to pick the best bets as you go through life. So let's say as a PhD student or earlier than that, you don't know much about anything, right, in terms of the depth in the field. So you take risks, but if you make it clear to your mentor and advisor that you're interested in challenging problems that will make an impact to the field and make a difference to society, then you can use the help of the mentor and so on to say, I'm willing to take risk, but I need help from you, right? And as you go along in life, let's say when you create your own laboratory and so on, you continue to take risks that now you're responsible for, but you also know that you have now dependents. On the home front, you might have a family and so on. So you can't take such a crazy risk that you lose your job. But on the other hand, what I really mean is that now you have mentees, students who are depending for their career in your life. So you have to, you cannot take so much risk that you destroy someone's career along the way. So your estimation of risk and what you do is based on what do I do to take the right amount of risk to enhance this person's career, and together can we mitigate this. So I've always said to people, take on always more than you can chew. That is, if you take on projects, instead of doing one and having it fail or succeed, take on 3 of those. At least one should be bread and butter, that you can get something concrete out of it, and then take on something more risky that will really make an impact on the field and answer a really challenging question. And then if it turns out that that risky thing is working, put all your effort into that, because then you know that that's going to work. So here's an interesting insight. So if I look back at the careers of my students, the most successful ones, and I don't want to, I want to use a very broad definition, is the most successful doesn't mean those who make the most money or whatever, but broadly, most successful and satisfying ones are those who actually struggled with their projects. So in contrast to those for whom everything worked while they were in doing their PhD, like a charm, and they published things that got out very quickly, they've been, in my view, somewhat less successful than those students who struggled and ran up to a brick wall and then figured out on their own how to jump that brick wall. And then they were unstoppable, meaning that they then went into different fields, into different disciplines, and made their mark, whether it is in biotechnology or in academic pursuits or in law, whatever it is, so they've made a big difference. So I think it's important to have, you need to have challenges in life, and you can face those challenges only if you're willing to take risks. So if you're conservative and don't take risk all your life, then you won't have any challenges, and you learn from these challenges. That's what builds resiliency in your thinking. And very importantly, it's also a key thing to learn how to ask the right question. So if I ask you, how does the universe operate? Very hard to answer that because it's a zillion questions in there. But learning how to ask the right question that you're able to tackle in the laboratory setting experimentally is a big part of your ability to answer the question. So that's also an important part of it.
So now this brings us to the most important part of research, how to handle failures.
Yeah. So I don't think that there's any real thing as failure. Yeah, if you, and let me just define that a little bit more. So if you screw up in doing something, that is, you didn't do your hard work and due diligence in tackling a problem, then I would say, yeah, the failure is lack of responsibility on your part. And for that, you just have to shape up and put in the hard work. But when you tackle a problem, there is no such thing as a failure if you put in the right effort, because you always learn from those failures, right? So, just to give you a funny story, when I finished my PhD and came to San Diego as a postdoc, I was very interested at that time in gene targeting. That is, targeting genes using homologous recombination to replace mutant genes, because I was interested in human diseases and the ability to cure it, and armed with the technology of being able to transform DNA into cells, this seemed like the obvious thing. And along the way, we got sidetracked into protein targeting, and so people would make fun of this and say, lost track somewhere and you move from gene targeting to protein targeting. But these are the kinds of things that it's not that the work that I did in gene targeting or DNA repair or recombination was a failure. I learned a lot from that. And now with the things that are happening at TIGS, it's come back full circle, because here after 40 years, the same questions that existed 40 years ago still exist today. What is the mechanism of homologous recombination in mammalian cells? How do you target DNA to specific places? How do you edit genes in place? So things have a way of coming back in circles. So I don't believe that if you keep your eyes and ears open, that there is any real failures in trying to address a particular problem. It often might be that you are too early and don't have the right tools to answer that, and you just have to keep that interest going so that you might come back to it at a later point. And there's always things to be learned from these so-called failures, and that's what makes you a better person.
Yeah, I think I would like to say that this is what makes you a visionary. Professor Subramani had a question 40 years ago, and now half of the world is trying to understand that, and with the available tools, trying to solve that question of DNA repair, or doing the gene drives, or curing the human diseases, like now people are doing for cancer and for sickle cell anemia, that you can do the CRISPR and do that thing. This is what makes you visionary. You might be ahead of your time. So we would like to tell every researcher, don't think that you are thinking of something too advanced in your career or whatever the tools we have available today, because your vision can actually lead some people to develop those tools. So always follow your questions. And as Dr. Subramani said, ask the right questions and keep pursuing your passion. If you're passionate enough, research is for you, for always. And with this, we are running short of time, and we would like to ask our final word of wisdom, Professor Subramani, what you have to convey to our researchers all over the world.
So I want to put out a set of general principles, that I want the young people to think about. And I've mentioned these along the way. The first, look for an untrodden path. That is, don't go down the same path that someone else has done. Look for untrodden paths. The second that I've emphasized is follow the question, the biological question, and don't get so enamored by the tools. The tools ultimately are indeed a tool to get you from A to B. And so don't get enamored by the tools, and learn to ask the right question that can be answered with the tools available at that particular point in time. The third is science is not just about you. It is about your ability to interact, collaborate, and most importantly, keep learning all through your life. Right? And fourth is you need to be passionate. That is, you need to live it, dream it, and so on. And it just takes hard work to stay ahead of the curve. But I've never viewed this hard work as something that was a burden on me or that was imposed on me. I've just had fun doing it. And so, you think of something that you really like to do, whether it's cooking or hiking or photography or whatever, that doesn't seem like work if you really love it. And that's what I want to push. And then finally, I just want to give some general philosophical advice that's true for any discipline. First, always try to give back more than you take in whatever aspect of life you're doing, whether it's from your students, from your administration, from the university employer, whatever it is, give back more than what you can take. And Raja knows this because at TIGS, I've always said TIGS should be known as an entity that gives back more than it takes. I'd say I had one foot in academia, one in industry, one foot in academics, one in administration. So at any point, I felt like, okay, if this doesn't work, I always, So the next point I want to make is that you should always create a fork in the road for yourself and not wait to be surprised by a fork in the road as you take a turn. So in other words, what I mean by this is that I've always had at least two alternative paths in my career. Some at times in academia, at times in industry, at times in basic lab research and administration, and doing things for society. So by having this, you mitigate the risk that you take. You can take more risks as a result of having an alternative path, and you can, as long as you have the confidence that you can move into another field and make an impact in there, this really helps you. And the final thing I want to say is that, you know, this is a long race that you're in for. And as life expectancy grows, you know, it's very unlikely that anyone who is getting trained in any discipline is likely to stay in that discipline for the next 40 or 50 years. So it's all about learning. And if you think of this as a race, I will just say that races are often won on turns. You know, there will be long stretches where there's not much you can do. But if you're able to make the turns with agility and with awareness and with acknowledgement of help from the rest of the people, I think you can turn your life into something really meaningful by serving society and helping people broadly. So that's the message I have. And I wish everyone the best of luck, and keep up the hard work and enjoy yourself mostly. Yeah.
Thank you very much, sir. We cannot thanks enough to you for taking your time out. And I'm 100% sure that the researchers all around the globe will be benefited from your enlightened word. And thank you very much.
Yeah, thank you. Have a good day.
Yes.